Hello friends, welcome to Bionie Nirmal, and we were talking about the Drosophila development, and mainly we were talking about the development of anterior and posterior axis and the dorsal and ventral axis of the Drosophila. Okay, and if this is an adult Drosophila, and this adult Drosophila contains the three main regions. One is the head, another is thorax, and this is abdomen. Okay, so this is head, this is thorax, and this is abdomen. So head and thorax is the anterior region of the Drosophila, and abdomen is the posterior region of the Drosophila. And we are talking about that how this anterior and the posterior regions becomes patterned. If this is a body of Drosophila, so this is the anterior and posterior axis. Okay, and these are the anterior and posterior axis, and these are dorsal and ventral axis. So this is dorsal and ventral axis, and this is anterior and posterior axis. And we are mainly talking about that how the anterior and posterior axis and the dorsal and ventral axis are specified. Okay, and also in the previous video tutorial I have described you about the process of meriostrichogenesis and also the formation of syncytial blastodome and the cellular blastodome. Okay, but this patterning actually started during the time of the oogenesis. This is very important. And in the oogenesis, you all know that the egg chamber is developed. Okay, and in this egg chamber there are presence of many of the uncommitted polar follicle cells, and in this video tutorial mainly I shall show you that how these uncommitted polar follicle cells becomes anterior follicle cells and posterior follicle cells and dorsal follicle cells and ventral follicle cells. So what kinds of genes are actually involved for development of the anterior follicle cells and posterior follicle cells and dorsal follicle cells and ventral follicle cells? There are presence of many of the nerve cells, and there are presence of 15 nerve cells, and just behind these nerve cells there are presence of oocyte. Okay, so Drosophila's anterior posterior axis is specified during the time of oogenesis, and this specification started by the movement of the nucleus towards the posterior region of the oocyte. And you can see that the nucleus actually moves towards the posterior region of the oocyte, and just after the movement towards the posterior region, this nucleus produces many of the proteins, and these are called as the Garkin protein. And you can see that these red dots are actually the Garkin protein. Okay, after the formation of this Garkin protein, the terminal follicle cells. So this is the terminal follicle cells, and this and after the formation of the Garkin protein by the nucleus or the oocyte nucleus, this terminal follicle cells produces the torpedo receptor, and this Garkin protein binds with this torpedo receptor, and this will causes the development of many of the microtubule. And these microtubule are very important for development of anterior follicle cells and the posterior follicle cells. Okay, and you all know that in microtubule there are presence of two regions. One is non-growing end and another is growing end, and I have denoted this non-growing end as the minus sign and the growing end as the plus sign. Okay, and in the non-growing portion there are present a kind of mRNA, and this is called as the bicoid mRNA, and this bicoid mRNA is actually the maternal gene product, right? Okay, and towards the growing portion there are presence of Oscar mRNA. Okay, and this bicoid mRNA is present at the anterior site of the developing oocyte, and these are very crucial for development of anterior axis of the Drosophila. Okay, and next this Oscar mRNA actually moves towards the posterior region of the oocyte. Okay, so towards the growing end of the microtubule, the follicle cells becomes the posterior follicle cells, and towards the non-growing end of the microtubule, the follicle cells becomes the anterior follicle cells. Okay, and next this nucleus moves towards the dorsal side, and next this nucleus moves towards the dorsal side of the oocyte along with its Garkin protein, and this will mix the follicle cells which are present above the oocyte nucleus into the dorsal follicle cells. Okay, and the follicle cells which are present through a long distance from this nucleus with the Garkin protein, these are developed into the ventral follicle cells. Okay, and thus by this way the Garkin protein and the nucleus, oocyte nucleus, is responsible for development of anterior follicle cells and the posterior follicle cells and dorsal follicle cells and in the ventral follicle cells. And you can see that in the oocyte there are present two important mRNA. One is bicoid mRNA and another is nanose mRNA. And this bicoid and the nanose mRNA are maternal gene product. Okay, they are during the time of Drosophila development, a series of genes are actually involved. Okay, so the first one is the maternal gene. So first one is maternal gene. Okay, and this maternal gene produces many of the mRNAs. Actually the nurse cells are actually present in the egg chamber, and this will produce the many of the mRNAs. And since these mRNAs are produced from the mother's Drosophila, so these are actually the product from the maternal gene. Okay, so these are not embryonic gene, but these are the maternal gene. And this maternal gene produces two kinds of mRNA. One is bicoid and another is nanose. Also hunchback mRNAs are also produced from this maternal effect gene. So this nurse cell produce the bicoid mRNA and the nanose mRNA, and these are the maternal gene product. And this bicoid mRNA are actually deposited towards the non-growing end of this microtubule. Okay, and this makes a gradient from anterior to posterior region, and this is called as the morphogenetic gradient, and this will determine the anterior oocyte into the anterior axis. So this is very, very important. And also the nanose mRNA, this will determine the posterior region of the oocyte into the posterior axis. Okay, so this maternal effect gene activate the gap genes. Okay, so this maternal effect gene activate the gap genes, and gap genes are responsible for development of these broad gaps, that is the head, thorax and the abdomen. And you can see that there are presence of the gap, and these gaps are actually developed by this gap gene. Okay, so these gap genes also activate pair rule genes, and these pair rule genes activate segment polarity gene, okay, and this segment polarity gene activate the homeotic gene, right? And in the next video tutorial, I shall tell you that how this maternal effect gene actually determine the Drosophila's embryonic cells into the anterior and posterior axis and the dorsal and ventral axis, and also the function of the gap genes, pair rule genes, segment polarity gene and homeotic genes. And these genes act sequentially, and this will causes the development of the structure of Drosophila's embryo. Okay, so these are very, very important, and you should know that what is the function of these genes. Actually the product of these genes are transcription factor, that is the maternal effect gene produce the bicoid and the nanose, and these are the transcription factor, and this will activate the gap genes. Okay, this will activate the gap genes transcription, and also the gap genes proteins are also transcription factor. This will activate the pair rule genes, and the pair rule genes are also transcription factor. This will activate the segment polarity gene. Segment polarity genes proteins are also the transcription factor. This will activate the homeotic genes. So this series of the genes actually acts on the Drosophila's embryo. So in this video tutorial, you can understand that the Drosophila's development actually starts during the time of oogenesis. Okay, and during the time of oogenesis, the anterior oocyte which is accumulated by the, and where the bicoid mRNAs are accumulated, and these are specified into the anterior axis, and the nanose mRNA which are accumulated at the posterior region of the oocyte, and these are developed into the, and these regions are actually specified as the posterior axis. And also the nanose and the Garkin protein and the nucleus along with the nanose and the Garkin protein, and this will transform the oocyte into the dorsal site, and beneath of this region this will produce the ventral site. And here I shall tell you that how these dorsal follicle cells and the ventral follicle cells are actually developed, and this oocyte nucleus along with its Garkin protein actually moves towards the dorsal side of the oocyte, and so that this region becomes the dorsal follicle cells, and the follicle cells which are actually present from a long distance from the oocyte nucleus, and these are developed into the ventral follicle cells. right? So in this video tutorial, you have learned that the anterior posterior axis is specified during the time of oogenesis, okay? And in the next video tutorial, I shall tell you that the function of the maternal effect gene and how the bicoid, hunchback, nanos and the caudal proteins actually determine the anterior posterior axis of the Drosophila and also the dorsal and ventral axis of the Drosophila, and also the terminal genes actually specified the terminal portion of the Drosophila, okay? And also the functions of the gap genes, pair rule genes, segment polarity genes and the homeotic genes. So thank you, and that's it.